Thank you all so much for coming and welcome to the UW-NPS Research Station at AMK Ranch. We are here for the 2024 Harlow Summer Seminar Series, which is a series of events that we hold throughout the field season while the station is open, and we have talks on lots of different topics, and we're really, really pleased to have you all here with us. So thank you for coming. And just to give you sort of a light summary of the events this summer, we're going to have seminar talks like this one on sagebrush restoration, on the tumultuous history of Sargent's Peninsula, which is the land that we are on now, on photography in the American West, on great gray owls in the greater Yellowstone ecosystem, and on alpine stream research. So those will all be really wonderful. And then in addition, we have the Wyoming Festival, which is the chamber music event that we have every year. Composers compose new music that's inspired by the greater Yellowstone ecosystem, and it's a really special evening. And then we're having a full weekend day outreach program this year that is focused on birds. That'll be with WyoBird, so that'll be a full day of activities, including bird walks and banding demonstrations, and some talks on bird research as well. So we would love to have folks up for those. So I just want to plug those as well at the start of the season. And then without further ado, I want to introduce today's speaker. We have the pleasure of having Dr. Willie Fetzer give our first talk of the season today. Willie is an assistant professor in the Department of Zoology and Physiology at the University of Wyoming, and also a member of the WYACT project, which is Wyoming Anticipating Climate Transitions, which is a really wonderful EPSCoR grant with NSF that we have a lot of really wonderful researchers working on. Willie's research focuses on how human-driven changes affect freshwater ecosystems, including the productivity and stability of Wyoming fisheries. And before Willie came to the University of Wyoming, he was the Great Lakes fisheries specialist for the Wisconsin Department of Natural Resources. And so I'll transfer things over on the computer and then let Willie start talking.
Thank you, everyone. Thank you, Hillary, for welcoming me for the first seminar. It's really exciting to be here. And yeah, what I'm going to talk about is really an overview of the research and how my lab thinks about fish communities and how they're responding to global change. Things like climate change, changes in the hydrologic cycle, changes in nutrients and how they're getting delivered to ecosystems. And so this is really like not so much a focus on any specific thing, but sort of how we think about fish community change at broad spatial scales. And so as Hillary mentioned, prior to moving to Wyoming, I was a Great Lakes fisheries manager for the Wisconsin DNR. And a lot of what I did and a lot of what other people I worked with was trying to understand how fish communities were changing in this landscape, right? Changing in the Great Lakes, in the largest lakes. 20% of the Earth's fresh water, right, is right in this picture. But also, what about streams, right? How are stream fish communities responding to environmental change? How are lake fish communities responding to environmental change? And how do we develop a framework so that we can sort of share ideas about how fish are changing across these different ecosystems and see like, okay, yeah, what you're telling me is consistent with what I'm seeing, or maybe what's really exciting is when we see differences, right? So we have this framework for how we think fish communities should be responding, and it actually allows us to identify places where it deviates from our predictions, right? And that's really valuable. But the first thing we have to do is have some sort of shared framework. And so has anyone ever been in this picture before? Has anyone ever not been in this picture before? Really? That's amazing. So you can imagine, right, that, there's a diversity of lakes. There's a lot of water in this picture. A lot of the team WYAC people were just in this picture a couple of weeks ago. And it's really different. Like you feel just so different. There's so much water. There's water in the air. There's water on the land. There's mosquitoes everywhere, right? But so can we take what we learned here and can we use those lessons and better understand Wyoming fisheries? And so as a young fisheries scientist, about 20, 30 years ago, this is what the framework was, right? This is how you should be thinking about fisheries management. How do fish communities respond to ecosystem change? And so, right, you had a lot of people, you could say, I'm going to study fish, right? I'm going to study habitat. I'm going to study people. And so when you look at this landscape, right, can you show me where there's just fish and no habitat? Anyone? I know you can't do it because you've never been there before. Can you show me where there's habitat and no fish? right? We're anywhere where there's no people, right? So this is a really bad framework to teach students, right? Because the fish don't exist without the habitat, and the habitat is all being affected by the people, right? And so this is a terrible framework to be teaching students, right? Really what we want to be doing is thinking about it at the fish first. And so we can learn things about the fish. We can study what their reproductive biology is, right? We can study what their thermotolerance is. What's too cold for the fish? What's too hot for the fish? We can understand how the fish feeds, right? Different fish, some fish feed on the bottom, some fish feed on the top, some fish eat other fish, right? Big fish eat the little fish. But we can study the fish and we can understand the fish. But we do that because we want to relate it to the habitat, right? We want to understand what's happening with the fish because we want to understand how they're responding to these habitat changes, right? And so when we teach students, when we teach the public, when we talk about them, we don't want to have fish over on one side and we want to have habitat on the other side, right? The fish are always within the habitat, right? So that's step one, right? We want to integrate these within concentric circles because we don't want to separate these things. And then finally, the people are sort of always around, right? They're always affecting it. It's even if you go to the winds, the top of the winds, and you find lakes that you feel like are very pristine, they're still being affected by nitrogen deposition, right? They still were potentially stocked with fish 100 years ago or something like that, right? And so we can't separate these things. So why should we even try, right? So step one for your framework on how to understand fish community change is we don't want to separate fish from the habitat from the people. Right? All of these things are connected all the time. Okay, you guys got that? So what does that look like, right? And can we develop actually some unifying hypotheses that allow us to think about how fish communities are going to respond in lakes, in streams, in the biggest lakes on the planet, right? And so first, right, or actually my wife, Sarah, keeps telling me that I keep saying right during talks, and so I'm going to try to work on it today. So the first hypothesis that we really want to start with the fish. And what we would think is that as the environment changes, species with shared traits should respond similarly to ecosystem change, right? So if you're cold water adapted, you should respond to temperature changes the same way as other cold water adapted fish. Does that make sense, right? If you are, a lot of fish have low oxygen tolerance, right? They can tolerate very low oxygen. So these are fish we think might be tolerant to eutrophication, or really high nutrients, very green water. Whereas fish that are very sensitive to low oxygen might be very insensitive to eutrophication, right? So they're very intolerant to very green water. They need to live in very clear water, right? Anyone think of a fish that might be?
A trout.
A trout, yeah, perfect. Whereas things like carp, we might think be more adapted to warm water and being able to tolerate low dissolved oxygen. And so with this first hypothesis, right, we can think about how individual species should be responding to environmental change, right? Species that are cold water tolerant, that are very sensitive to low oxygen levels should respond fairly similarly to environmental change. Other species are a little bit more in the middle, right? They can handle cold and cool temperatures. They can tolerate a range of dissolved oxygen levels, right? They're likely to respond similarly to environmental change. And then there's also a lot of species like catfish, right? They're super tolerant to low oxygen levels, right? They don't mind warm temperature at all, right? And so we would expect that across these different ecological guilds, or these species that have these shared traits, that as the environment changes, they should respond together, right? That's our hypothesis. It doesn't mean that that's what happens, right? And when it doesn't happen, that's what's really exciting from a scientific perspective, right? But this is our sort of baseline underlying principle, right? Our underlying hypothesis is that species with similar traits should respond similarly. And if that's the case, then as the environment changes from being cold and oligotrophic, or cold and very clear, very low nutrient levels, to being warm and eutrophic, so having lots of nutrients, very green water, that these fish assemblages should actually form a continuum, right? And so as you move to being warmer and having more productivity, greener water, you'll lose fish, but you also pick up other fish, right? And so it's not that you're just gonna like lose all these fish as the environment changes. Other fish should be adapting or should fill that place if they're able to move, right? This is sort of a hypothesis that we can test. And because individual fish are shifting along these gradients, even though the environment changes, we would hypothesize that fish biomass and production should be maintained fairly stable. Does that make sense or not? Yes? Yeah, and so like when you think about this, if you have a cold oligotrophic system, something like this out here in Jackson Lake, you can identify what species you would expect to find there, right? Because these are those species that are cold tolerant and intolerant to low oxygen. And as we move and we move along environmental gradients and we get towards warmer temperature with more productivity, we see that generally we have new species replacing it. So things like walleye and perch start to become more common. We lose things like lake trout. We might lose things like burbot. And as we continue along that gradient, we're gonna continue to see fish species composition shift, right? So we're gonna gain more and more species that are tolerant to warm temperatures as we lose those cold species. But along these environmental gradients of temperature and productivity, we expect the community to still be maintaining that production, right? As you lose one species, other species oftentimes are able to respond and maintain that production. And the challenge, though, is how easily can they move between those spaces, right? So fish species within an environmental conditions can adjust who's dominant and who's not dominant, but you can also have movement. And we see this a lot of times in lakes where we have long-term data sets and where we are able to track fish community change in response to environmental shifts, right? And so we can see that in a lot of places in the upper Midwest, the systems were really eutrophic, but environmental regulation, things like the Water Quality Act, the Great Lakes Water Quality Agreement have actually increased water quality, right? So the water quality has actually moved from here to here, and we see the fish communities respond. We see that those intolerant species come back. We lose some of the tolerant species. And so there is some capacity of these fish communities to reshuffle and maintain biomass and production, even as the species assemblage shifts. And then finally, ultimately, the capacity of fish communities to reshuffle this dominance and this composition is going to be what drives the resilience of the productivity to different ecological perturbations. So basically, what I'm saying is that the capacity of the ecosystem to move between these fish communities will determine how stable they are, right? Some fish communities, some lakes have a lot of different habitats, and so they can respond, and the fish community does respond really well to environmental change, where other systems, this feedback may be really difficult, and so there we don't see the resilience, right? The species are unable to reshuffle, dominance is unable to change, and so that's where you see these major shifts and you lose that stability. And so this is just a framework, and these are just hypotheses, right? But it's some way that we can think about how are lakes, rivers, or the largest lakes in the world changing, but can we actually see that when we look at it on the landscape, right? Does the data actually support this framework? And so we worked with the Michigan Department of Natural Resources, which is a really great agency. They have about 1/3 of the Great Lakes shoreline is all contained within Michigan. I think Michigan is the #1 state or the #2 state in the nation for coastal shoreline, second only to Alaska. So there's a lot of Great Lakes influence, and they do a lot of monitoring in the Great Lakes ecosystem. And they do different surveys. They have surveys that are along the exposed coastlines of Lake Michigan and Lake Huron. They also do surveys in coastal embayments, right? So these are places like Saginaw Bay and Badenoch and the St. Mary's River and the Lachino Islands. And so what this allows us to do is see how our fish communities structure across a really wide range of environmental conditions, right? Really cold, oligotrophic, very deep, to very shallow, very warm, extremely productive ecosystems, right? So do we see these patterns actually exist on the landscape? And so here you can see those sites. And what you can see here, right? This is just like that plot I showed you where you have the different species plotting. And you have essentially each of these sites is associated with a different fish community. And so if you're plotted here, that means that you probably have alewife, burbot, lake trout, whereas if you're plotted over here, these are things like walleye, channel catfish, quillback, and other like things like suckers and rough fish, I guess people would call them. And so what we see is across this landscape, we do see these really clear patterns in terms of how fish communities are structured, right? So when you look at exposed coastlines in Lake Michigan and Lake Huron, they actually have a very small space, right? So fish community is extremely similar across hundreds of miles of shoreline, right? Each of these plots is an individual fish community sampling event, and they're all almost entirely on top of each other. Right, so very, very limited fish community change, almost all the same species, very limited differences among sites. But when we look at our embayments, we see a huge range of fish community differences, right? We see, right, I keep saying it, I'm sorry. In Saginaw Bay, we see a lot of warm tolerant species. We see species that are tolerant to eutrophication. This is the ecosystem that has the warmest temperature and has really bad eutrophication problems. When we look at the Lachino Islands and Bay de Noc, right, areas that are sort of intermediate, we see these intermediate fish communities, things like northern pike, things like smallmouth bass start showing up, right? And so it's actually matching this prediction quite well, actually. So in the exposed coastline where it's cold and oligotrophic, we're seeing that fish community dominate there. Right, this is your classic Great Lakes fish community, lake trout, Chinook salmon, round whitefish. As we move into the embayments, we start picking up a cool and more tolerant species more tolerant to eutrophication, like walleye, smallmouth bass, yellow perch, crappies. And as we move into the warmest, most eutrophic areas, we lose a lot of those species and we start picking up more warm water species. We start picking up things like quillback and suckers and other species that are really tolerant to these environmental conditions. So it does appear in the Great Lakes that when you plot these parameters, when you look at how these environmental drivers like depth, productivity, and temperature, how they affect fish community, it seems to match that framework actually pretty well. Right, and so we can take these individual surveys and we can combine them to actually think on the landscape of who actually lives in which location. Right, we can convert what they're doing from their individual surveys and give them a holistic picture of how the fish communities in the Michigan waters of the Great Lakes are structured. Right, so in the exposed coastlines, you're generally going to find these cold oligotrophic system or fish communities. But as you increase in productivity and temperature, you see this shift towards these increasing dominance of warm water species and species that are tolerant to eutrophication. Right, so it seems like these shifts that we would expect are occurring on the landscape. And we can think about this again to sort of, okay, what does this all mean? Right, how do we combine this in a unified framework? And ultimately what we want to know is as you add more nutrients into the system, right, as that system increases in productivity, becomes greener from blue to green, how do we expect fish biomass to respond? Right, so if we add more nutrients, do people think fish biomass is going to go up? Yeah, right, okay, just making sure. Right, and we also think though that food web trophic transfer efficiency matters. Does anyone know what that means? So basically trophic transfer efficiency is how that food web is structured. So if you have a bunch of apex predators, a bunch of large lake trout or something in the system, it takes a lot more energy to produce those fish. So the total ecosystem production we expect will go down. Okay, whereas if you have a bunch of lower trophic level fish, you can produce a lot more because you're losing less energy as you move up the food web, right? So think about, right, big fish eat little fish. This is where this matters, right? How the big fish eat the little fish really has a big impact on how the ecosystem converts energy into biomass, right? Fish biomass is what we're talking about. So you guys probably remember, or maybe you don't, from 90% of energy is lost every time at each trophic level, right? Anyone heard that before? So if you have zooplankton eating algae, however much biomass you had, you'll lose 90% as you go up. And you go up another trophic level, you lose 90% of the 90%. And you go up again, you need 90%. So basically only 10% is conserved as you go higher and higher into the food web. And so if we have food webs that are structured like this, where you have very few apex predators, then you're going to have, and your fish community is dominated by second and third level predators. then you're going to have a lot higher biomass, right? Because you're having to produce less biomass at higher trophic levels. Does that make sense, right? Whereas if you have a bunch of walleye, it takes a ton of energy to produce top predators because it has to go from here to here, all the way up to here. And so in this type of food web configuration, right, if your fish community is structured where it's dominated by big fish, then you'll produce less fish.
Yeah, okay.
Yeah, right? If your food web is dominated by a bunch of suckers, you can produce a ton of suckers, right? Like you'll just produce a lot and a lot. So top predators are harder to produce, they take more energy. And so food web structure matters a lot too. With me on that? All right, and so what we can do then is start looking at these different sites. And we can see in individual locations as nutrients have changed, as temperature has changed, as all these different factors have changed, we can start piecing together what this might look like on the landscape, right? So it's not necessarily though, just this fish community A to fish community B, right? It's probably more something like this landscape. So that as nutrients increase, or things that are temperature or depth, that you would expect to see this transition in the fish community matching those functional traits, right? Those shared characteristics. So down on the bottom, you're going to have more cold and oligotrophic fish. As that temperature and that productivity increases, you should see that transition towards more cool and mesotrophic fish. And as you get to warm and eutrophic, you would expect there to see, again, that transition into these different fish communities. And what's really valuable is we can actually put the fish on there. And we can actually start talking about now, okay, well, what does this actually look like? Like, what the hell is Willie even talking about, right? And so the idea is that as nutrients increase, we expect fish production to increase, which we talked about, right? And we also expect the composition to change. And so we can see that cold oligotrophic community here. We can see that cool mesotrophic community here. You see that it's a little bit, you know, those spaces are less overlapping, right? So this is a lot harder transition. Like we don't think that the Great Lakes are going to turn into this fish community anytime soon without major ecosystem changes, right? Because this is a hard shift. Whereas in this area, like it's actually, we see fish communities move back and forth between that space all the time, right? When we've seen as the Great Lakes Water Quality Act has restored water quality, we see that the fish communities are moving back this way, right? So that's really positive. The other thing though is we can also incorporate into this what we were just talking about in terms of the food web structure, right? And so if you see on the bottom, anyone want to guess what's shared characteristic these fish all share? They're big. And what do you think they eat?
Fish.
Fish, yeah. So if you have a bunch of fish predators in your food web, then you will have less productivity, right? You would expect the fishery to be less productive if all you have is walleye. Whereas if all you have, if you look at these, right, these are all lower trophic level fish. We used to call them rough fish, is probably things you probably heard, right? If you have all lower trophic level fish, then in any given environmental condition, you'll produce a ton of biomass, but it'll be a bunch of carp and suckers, right? And so that's not necessarily desirable either. And it's really in this intermediate space where we probably have the best benefit, right? We have walleye, we probably have good prey fish, right? We have the most diverse fish community. But we can actually see like how environmental conditions are affecting who lives there, and then how these species interactions influence how productive the ecosystem is at that specific state. With me? And so we're actually starting a new project right now, and we're going to test this across the entire Great Lakes. And so you could imagine, right? Well, we've only looked right here and we've seen these really interesting trends, but this is Western Lake Erie, which is very, very green, extremely bad harmful algal blooms. I mean, it like when you talk like pea soup, this is the stereotypical picture. To Lake Superior, which is one of the least productive, very low productivity systems on the planet, right? Very large, very, very intact watershed, so very low nutrient loading, very deep, high, so really large range of environmental conditions here that we can test. Does this framework actually hold up? And so what we've been doing is working with state, federal, and tribal agencies and provincial agencies across the entire Great Lakes. And we've been able to put together over 40 fish community data sets to try to ask this question, like, okay, this framework seems to hold in Michigan, but does it hold across the entire basin? Here's just here's just an indicator of the extent of sampling that occurs in the Great Lakes in just one specific year. So you can see Lake Erie again gets a ton of sampling. Not surprising, that's where most of the people are. But again, Lake Superior, lots of sampling. Lake Michigan, lots of sampling throughout. We can sort of summarize this and see, just like the stars that I was showing you in Michigan, we have these stars all across the basin, right? And it actually looks exactly like this picture, really, if you go back and forth. And so what we're going to do is see, does this actually hold, right? Does this framework hold? We can look at not just these fish communities in specific locations, but we can look at it across the entire basin, right? And we can start communicating to stakeholders and decision makers and our agency collaborators how the fish community looks now, right? What does this star represent? We can demonstrate that with the fish icon, with a word cloud or a fish cloud, if you will. And also track how those things are changing over time, right? So we can show what happened in Saginaw Bay as the ecosystem recovered. We can show what's happening in some of these systems as new species are getting introduced. So we're really excited about that. It's sort of an ongoing project. But what helps me a lot with this framework is that currently, when we first started, if I said to you, well, how do you think fish communities are structured? Right, we probably would have got a lot of different answers. People would have interpreted the question really differently. But for me, this really helps because it just puts on paper what these relationships are in my head between environmental conditions and fish community across scales, right? It's something we can look at. You can take a look at this and you can say, I think you're wrong. And that's great, right? Because we kind of had that conversation before, right? So it puts it in a framework where we can sort of start saying conceptualizing, I guess, what do we all think? It also allows resource managers to develop their own hypothesis about how these different factors are interacting and affecting things like water quality that Sarah studies and fisheries that I study, and propose possible interventions to achieve their desired results, right? So this is what mine looks like, but they can say what theirs looks like too, right? They can talk about where do they want it to go and actually start having a more open dialogue and making sure that they're all on the same page in terms of what their management targets are and different actions they can take to get there. And then finally, what I like kind of building on that last point is you can maybe anticipate how these interactions between changing environmental conditions and management actions, things like habitat enhancement, right? Changing the habitat to try to promote fish production, stocking, putting fish in to try to add, like make more fish, or regulation. So this is how we harvest fish, right? This is how we kill fish. This is mortality. So mortality, birth, and the habitat quality, right? We can start thinking about all these different actions and how they might affect or shift that ecosystem in one way or another, right? So if this is where we think our fish community is now, we can actually have a conversation about how different management interventions like stocking or regulations or habitat enhancement are going to move that needle, right? And this is important because sometimes the fisheries people want to push it this way. Well, what do you think the water quality people want to do?
Push it the other way.
They want to push it this way, yeah. And so in the Great Lakes, this is actually something that we're confronting. Like the Great Lakes Water Quality Act has worked. Water quality is better, but fish production is down, right? And so this is something that environmental regulators are having to deal with and think about. So this is sort of something that we've been building in the Great Lakes, where there is a lot of data available, where like the Great Lakes are really nice because they are contiguous. So a fish could move into Lake Superior, right? It's a lot harder in lakes and streams where you have dams, or maybe the fish can't move from one lake to another, right? But can we adapt this framework to large lakes and reservoirs in Wyoming to better understand what's happening here? And so one thing we can see is that in these reservoirs, right, these aren't lakes that were naturally here. Like this was a natural lake, but a lot of these large lakes were very limited in Wyoming, like limited to the winds on either side. And that's about it. All these other large systems are reservoirs. And so just from a game and fish perspective and from a regulator, we've already used this framework with where we've stocked fish. Right, we see that the apex predators in the lower elevation reservoirs are dominated by walleye. Right, as we move further down, we start picking up bass. In some of these places, we start picking up northern pike. Right, so the managers are already using this framework in terms of where they're stocking fish historically. Right, and we see lake trout only in these most high elevation systems. But in Wyoming, because it's so much is manipulated by the managers, because these systems, they are these reservoirs just in the middle of the desert, and so a lot of the fish community that there has been put there by us, right? And so a lot of what's really driving the productivity in these systems is less about the ability of the community to respond to environmental change, and it's more about how fish are eating other fish. Right, so in this system, the environment's having probably less of an effect because the people component is so high that the fish then, though, are really having effect on what the outcome is, right? The food web structure, who's eating who, really seems to matter, we think. And so what do these systems look like? Well, in most of them, they're basically walleye and lake trout, which are naturally reproducing. So we don't, like the game and fish is not stocking these anymore in any of these systems. And they're supported by a mix of either naturally reproducing native fish that were originally in the river ecosystem. Some have potentially been introduced accidentally. And then some component of stocking of trout, like or kokanee salmon or Bear River cutthroats, right? So these food webs are being maintained both by natural reproduction of prey fish, but also a lot of stocking by the Wyoming Game and Fish Department, okay? And their management goal, and what they've been actually really successful for the last 30 or 40 years, is maintaining abundant trout while also having trophy predators, right? So you can think about that in the framework. They're sort of in that middle space, right? Where they're not too dominated by apex predators. So they're able to have people can go catch big lake trout, right? 50-pound lake trout on Flaming Gorge, 40-pound lake trout on Jackson Lake. But they also can go catch a bunch of kokanee, right? They also can go catch a bunch of rainbow trout. And for a long time, that was actually working really well. Unfortunately, though, what we're seeing is increasingly many, many more small predators, right? If anyone has heard all the conversation over the last year or two about Flaming Gorge and trying to harvest pups, right? The Game and Fish is turning, as I think successfully, or is trying to successfully pass legislation to make lake trout a non-game fish so that people can just throw them away because they're so concerned about how the food web structure is changing, right? And they're seeing consistent stocking failure. So they put in 30,000 pounds of rainbow trout in the fall, and they go back in the spring and they're gone, okay? So this is a real problem for them, all right? It's a real problem for us because we want to go catch big fish. People want to come here and spend their money and catch big fish. And here you can just see here's some of the press. Flaming Gorge Reservoir anglers should expect reduced kokanee salmon catch rates. Concern over Flaming Gorge lake trout. They're really pushing like very liberalized harvest regulations, so high bag limits, because they want to reduce the population of these fish because they're so concerned about the food web structure. And sorry, just laughing because we show this food web structure, right? But it's actually so complex, right? Who's eating who out there? What big fish are eating what little fish is really hard to figure out. So even like, here's sort of what we might see on Instagram. These are some extremely famous lakes in limnology, in the study of fresh water. They're very small. They're probably maybe 5 or 6 times as big as this room. And when we study them, we describe the food web like Instagram, right? Like a meme or something, where there's algae eaten by zooplankton, and then a fish eats them, and then a bigger fish eats them, right? And so we develop these really simple models of how the food web looks, but in reality, this is the food web of this lake. And so it's really like, okay, so how are these changes affecting prey fish, right? How are stocked fish interacting with naturally reproducing predators in Flaming Gorge? Is a hard question if this is the food web of this tiny little pond, right? And so one of the great things that's come around in the last 20 to 30 years is the use of stable isotopes. And this is food web analysis for dummies. So people like me can actually do this stuff now. And what it allows us to do is basically at the base of the food web, carbon isotopes are setting up different pathways that we can track as those pathways move higher up the food web with nitrogen isotopes, right? So we can take this extremely complex picture and we can simplify it into a two-dimensional picture or three-dimensional picture and actually see how all these species are interacting with each other, right? And we can ask things like, what does the food web structure look like? Where do yellow perch get their energy from? Who eats yellow perch? What do yellow perch eat, right? We can start asking those questions. We can ask things about the food chain length, right? How many trophic positions are in this ecosystem? How are fish partitioning the ecosystem? Logan back there is trying to understand how these stocked trout use the ecosystem after they're being stocked, right? Who's more vulnerable to which predator? We can think about ontogenetic feeding or how fish feed as they get bigger, right? Think about your kids and how they feed when they're little babies to when they get older. The fish do the same thing, and we can actually track this with the isotopes. We can understand how energy moves across ecosystems. We can understand omnivory, right? Because most fish aren't just eating one thing. And it allows us to better understand how these ecosystems are changing, right? What is the impact of invasive species? Who eats them? Who do they compete with? What happens when trophic state or the productivity changes, right? Where does the energy come from, right? We can track this stuff. We can understand things like climate change or introductions and extinctions and management, right? And so we can see how the food web is responding. Because it allows us to take this really complex reality in this tiny little lake and describe the Lake Michigan's food web in a way that's really easy for me to understand. Where we can see, right, here's the trophic position. The lake trout's at the top. Here's an offshore energy pathway. That's sort of algae that's floating in the middle of the water, right? That green that you see. Here's a benthic energy pathway. So that's plants that are growing along the bottom. And so we can actually see how the food web is integrating across these two carbon sources to support these apex predators, right? So we can collect the fish, we can run some chemical analysis, and allows us to take this really complex plot and visualize how Lake Michigan has changed in the last 20 years, which is a really big, complex lake. With me on that? And the other cool thing is that stable isotopes turn over at different rates for different tissues, right? So historically, if we wanted to know what a perch ate, we would have to look at the diet. But that only is going to tell us what that fish ate for the last 12 to 24 hours. And also, we can't tell what the fish ate the day before, right? So we can't track the same fish. We don't understand how fish are feeding across time. But if we use blood, we can run isotopes on the blood, and it can tell us a little bit longer window of how that fish has been feeding. We can run liver, and your liver is very metabolically active, so it turns over a lot, right? And so it's going to show that signature show up. We're going to be able to track how that fish is feeding at a little bit longer, but not too long of a time scale. And then muscle and bone have really long turnover rates, right? So we can see how this fish has been feeding for the last month or year, or if a 40-pound lake trout, it could be years that isotope signature represents. The other thing that we've been doing that I don't have time to talk about that's really cool is we've also been developing technology where we can actually take a fish's eye lens out, peel it like an onion, and run each individual layer and recreate that fish's feeding throughout their entire life. So we can go all the way back, and at the core, it's the mother, right? You think of like a fish when it's born, its mom had she deposits the egg, all that energy is coming from the mother. The fish, like if you ever go to a hatchery, the hatchery people talk about the fish eyeing up, right? That's an indicator that the fish is going to survive most likely, because the fish, when they hatch, they have to be able to see. So we can basically, with this eye lens technology, figure out what the mom was eating when she produced the egg, and then track individual layers to see how that fish has changed over the course of their life. And so we've done this a lot, and every time we do it, it's super cool. But a really unique example is we have a collaborator who works on the Arctic Circle, and she works with First Nations tribes in Canada. And what they have is these massive Arctic char, like just a beautiful fish. And what they do is they spawn in the Coppermine River and these other rivers, and then they migrate out into these estuary ecosystems because they can feed and grow a lot faster, right? And so they've been able to show this movement by catching fish in these offshore habitats. But what we can do is we can also show that with the eyeballs. And so here, like the way that you would interpret it is that at the very, on the left here, that would be the core. That's the signature that the fish is getting from the mother. And as we move out, those are sequential layers on the onion, right? So this is the outermost layer. This fish is growing and growing and growing. And we can see with the sulfur isotope that during their very early life history, the fish have a signature that reflects the freshwater ecosystem. As those fish grow and they transition, we see the island's isotopic signature transition. And now we can see that these fish are feeding in this marine environment, right? So these isotopes are really powerful because they allow us to figure out what habitats fish are feeding in, right? We can tell how this fish is feeding in the ocean or in the river, right? Really clearly. We can see that with carbon as well. Sulfur is really good for the ocean. Carbon's really good for the lakes. And so the carbon, what's really cool is you still see this transition, but what you can see is in that freshwater ecosystem, there's a lot more carbon diversity, right? The carbon isotope signature is much more diverse, but by looking at them together, we can really start to understand this fish's migration from these river ecosystems out to the estuary where they go and they feed on marine food sources, right? We can also do it the exact opposite way here in Wyoming, right? And so Game and Fish is really interested when they stocked fish at figuring out, is this a stocked fish or is this a naturally reproduced fish? And so we had the opportunity where they had actually clipped some fish and stocked them, and there were naturally reproducing fish in the North Platte River. And so they went out and collect them, and what we could see is that, so again, right, thinking as you go from left to right, this is from the core moving to the outer end of the layer, or the eye, is that when these fish were in the hatchery, they looked like the ocean. Because what do we feed fish in hatcheries? Ocean fish, ground up in the pellets, right? So when the Wyoming Game and Fish is raising fish in hatcheries in Wyoming, they look like they're swimming in the ocean. And when we stock them, we can see the islands as transition, and they start looking like the Wyoming reservoirs or the Wyoming rivers, right? So this is really cool, right? We can see it both going out into the ocean, and we can see when we truck productivity from the ocean and we turn it into little pellets and feed it with fish, that the fish show it too. And what's really, because these stocked fish look like the ocean, because they're fed a marine feed, they isotopically look marine. Here you can see, here's sulfur on the bottom, carbon on the top. These are, all of these are fish that are natural in Wyoming reservoirs. So this is what their signature would normally look like. In Pathfinder Reservoir, in Flaming Gorge Reservoir, in Jackson Lake, this is what they would look like. And these stocked fish, though, are way higher, right? This sulfur signature, you can see, it persists all the way over winter. So these fish are stocked in October. In June, when we collect them, they still look like the ocean. Carbon is not as strong, and nitrogen isn't as helpful. But what this allows us to do, and well, what if I do this? I probably should have done this a long time ago. We can use the sulfur stable isotopes to track what happens to these stocked fish. Because this sulfur signature is so unique, when we dump 30,000 pounds of sulfur into the lake, we can see where it happens to it, right? We can see who eats it. And here you can see that. So here's just carbon. This is, the black is muscle. The open circles are liver. Remember, muscle turns over slower than liver. Liver turns over really fast. So here you can see, here's what they look like. Here's what walleye looked like. Sorry, these are apex predators. And so we're trying to see if we can detect consumption of stocked fish, right? The stocked fish are up here. Here's what the natural ecosystem looks like. When they stock the fish in November, In Alcoba, we see that the liver does move up, right? In both of these systems, the liver moves up very quickly. If you can see that, right? It starts approaching this stocked fish signature. We don't necessarily see it super strongly in June. Part of that is because we think that the fish are just eating too fast. Like they stock them and they're just gone. And so they're not actually turning it into much biomass. The sulfur shows that much stronger. So here, right, see how high this liver moves in just a month? Like 10 parts per thousand in isotope terms is huge. Like that's a very huge shift, right? And so what we're seeing is that this is before stocking. When they stock the fish, we can see the isotope signature of the tissues start looking like the ocean, right? The walleye livers and the walleye eggs swimming around in Wyoming look like they've been feeding in the ocean because these stocked fish have such a high sulfur signature, right? So they're eating them, right? They're having a major impact on them. These stocked fish are an important prey source in these North Platte reservoirs, right? We see similar patterns when we look at Flaming Gorge. So here you can see small, medium, and large sized lake trout from Flaming Gorge. Not to distract too much, but these are the stocked fish, right? About over 50% of the diet is comprised of stocked fish, of these really large fish in these systems. Also important for the smaller fish too, which is important because numerically they're way more abundant, right? Hundreds of mouths eating a little bit at a time can have as much of impact as a few big mouths, right? And here you can see that even stronger with the isotopes. So this is estimated from the isotopes and from different models that we use. Again, really, stockfish are a very important component of these diets. So it is like, you know, it's good that they're stocking these fish. It's having a positive impact on these predators, but the challenge is they're not meeting the trout objectives, right? We don't just want walleye. We don't just want lake trout. And we also see this pattern in New Fork lakes. So again, less clear to see, but here you can see rainbow trout and kokanee in this purple are important diet items in New Fork lakes as well, right? So much so that my student Caroline was basically able to reconstruct almost 100% of the mortality of that stocked fish, right? So they put in 40,000 kokanee, and we basically can model that the lake trout are eating about 35,000 of them, plus or minus. And you know what? And where's Joe? And they get about 40,000 back at the weir. So we're pretty close. Like they're really important. These trout are supporting these apex predators, but the challenge is like, is this actually what we want, right? Are we seeing the impacts of these predators dominating the fish communities? So if this is where we want it to be, where we do have lake trout, we do have walleye, but there's also a bunch of stocked fish. And so the walleye and the lake trout we have are big and they're not super abundant, right? But we think maybe we're shifting into something like this, where these fish communities are becoming dominated by these apex predators. And it's really making it hard for the game and fish to manage and meet the objectives that they worked with the stakeholders to develop. And so how should the management weigh these competing paths forward? Well, one, they need to start thinking about, and they are thinking about, strategies to regulate and suppress apex predators populations, right? That's why John Walrath with the Game and Fish is having all these meetings in Flaming Gorge, because they're figuring out ways that they want to suppress these apex predator populations to support multi-species fisheries. They're thinking and they're assessing their hatchery capacity, and they're really creative and thinking about ways that when they stock to have it be more efficient, right? So that they're putting in fish, and it's not just leading to bigger walleye, it's also producing catchable fish for families and things like that. And also thinking about how do we manipulate habitat conditions to promote or resist fish community change, right? Are there things that they can be doing to try to fight some of these trends that they're seeing? I think it's hard. Like there's only so much we can do. Wyoming's a really difficult state because there's not a lot of people. And so in Wisconsin, if you had a bunch of walleye, there would be people catching them. But in Wyoming, like there's not a lot of people here. So it's really hard. It's really, if you liberalize the bag limits and you tell people they can catch 24 fish, but there's only 10,000 people that live in your county, that's going to be a hard sell, right? Their capacity is limited. And so I think they really need to be creative. And I think they are really creative. And the other thing is the other thing I like about this framework kind of coming back around is it allows us to think about what we might want to do. And so there's this idea in terms of how do we manage fisheries? We know that they're changing everywhere. We know that humans are having a major impact. And so how do we determine where do we resist this change? Where do we accept this change? And where do we try to direct it in a way that maybe matches what our goals are? And so a resist in this framework might look something like this, right? So if you're monitoring your catch over 4 years and it's fairly consistent, this might be a system that you think is resilient to change, and you want to try to resist any sort of shifts in the fish community, right? Alternatively, you might see a fish community, and we do see fish communities that look something like this, right? Where they're moving in this trajectory. It seems like they're kind of destined towards this more warm eutrophic fish community, that we should maybe just accept that, right? And let's stop putting resources in there. Let's stop stocking trout in that system and start thinking about stocking warm water fish, because maybe they'll do better, right? And we might also see like what we're seeing in some of these reservoir systems we hypothesize, where they're moving so hard in the direction that we might want to direct it back, right? So maybe this would be a good system where we would want to have a suppression program. We'd want to suppress apex predators to try to support more multi-species fisheries. But again, by thinking about it like this, we can actually see how the fish community is changing, think about what our potential management options are to get to where we want to be, right? And that can be the hard part, is like, where do we actually want to be in this fish community among a diverse group of stakeholders and practitioners? So what are some of the things we're going to do, especially like along these lines and with WIAC moving forward? Well, one is we really want to quantify how these environmental conditions are changing both within and across Wyoming. I mean, Wyoming is like, having come from, the Midwest with all this, there's so much environmental data. There's so much historic data available. It doesn't exist in Wyoming, right? And so it's a real challenge to sort of characterize how things are changing, but we really want to focus on that. So here you can see some idiot here in a raft looking at, and so we're putting buoys, right? Because we want to understand how the communities are, how the ecosystem's changing, excuse me, right? And what this allows us to do is compare places like Boysen Reservoir and Jackson Lake. And one of our techs, Kevin, put this together, and you can see just how much warmer Boysen is than Jackson Lake, right? You can see how much deeper that lake is mixing, whereas Jackson has a lot of cold water habitat, right? And so as we start understanding these changing conditions more, we can start thinking about what fish might be able to live there. We're also trying to, so that's just a buoy that's collecting real-time data all the time, but it's just in one spot. And so the other thing that we're doing is we have a new, with this WIAC project, we have a new boat. It's bright yellow. If you see it, wave and honk and harass the students because they hate it, and I love it. But what it does is basically we can drive at 20 to 30 miles per hour across the lake. And what we do is we pump water up onto the boat, we run it over sondes. And so now we can pair up this individual location with high resolution mapping across the surface of the lake. So how much does this point match up with that point, right? How important is the near shore relative to the offshore? And this stuff is very preliminary, but you can start seeing we're all like, so if this is Jackson Lake, just overlay it on top of here, you can see how the temperature is different higher up in the inlet. And as it gets down to the dam, it changes, and how dissolved oxygen across the surface differs across Jackson Lake, right? This is just one transect from the river down. We're also doing this with a raft. And so we want to understand environmental conditions on the Snake River, which essentially means that the students go on a raft, they bring their fly fishing poles, a picnic lunch. But what's cool is here is the start of Jackson Dam. And so you can see the temperature influence, but this is right about where the oxbow is. Groundwater or something is coming in. It very much changes the temperature, drops it down. The dissolved oxygen goes up, right? And so what we're going to be able to do with this is really characterize the heterogeneity of the Snake River, right? And how do tributaries affect it? How does it change across the season? And my sense is the students are very eager to collect a lot of data this way. right? But we can see these really cool trends. We can connect these point estimates with larger spatial resolutions and start thinking about water level changes and changes in the hydrologic cycle. And how is that going to affect available habitat for fish? How is that going to affect energy pathways supporting these different fish? So it's, we're really excited about that because this is a real major unknown in Wyoming. We also want to try to link environmental drivers with fish community dynamics, right? What is our framework for Wyoming? What are we trying to accomplish as we go from low elevation to high elevation? Where is it working? Where is it not working? Right? We need to start talking about this conversation, and we are. People are thinking about this. And then finally, we need to expand our food web models to understand how this structure is influencing productivity and stability. And so this is a lot to take in. Tristan Bletchinger is in the back. You can ask him all about it. But essentially, we've just been talking about the predators so far, right? But these are all the isotopic signatures of all the prey fish underneath it, of the zooplankton and the other components of the food web. And so you want to see, how do walleye that have this much tighter niche, right? How do they affect food web stability? How do they affect the success of stocking relative to something like lake trout that has almost a shotgun blast approach, where they're just like, I don't know, I'm just going to do this and hope it works out. Where the walleye are just, they're all doing exactly the same thing, right? So how does that affect stability? How does that affect productivity? How does that affect the fish species capacity to adapt to management changes, right? Walleye might be really difficult to outmanage because they're so good at what they do. Whereas lake trout, maybe they're a little bit easier. We don't know. These are questions we want to try to ask. And so what I'll end with, which I'm sure you're all happy to hear, is that we're taking these ideas from the Great Lakes and we're applying them to Wyoming, and we're really learning a lot, and it's really valuable. But I also think that we're going to be able to send some really valuable information back there, right? Because we can manipulate these reservoirs in a way that you can't do so much out there, and we can see the importance of food web structure. We can understand, how having so many apex predators maybe isn't that good, right? Maybe that's not what we want. And so anyways, hopefully you guys learned something. Happy to talk more about fish. Again, this is just a framework for thinking about how fish respond to environmental change, and I'm sure it's not right. So with that, I'd like to just thank some amazing undergrads and graduate students who have done all the work, some great academic, state, federal, and tribal collaborators, some funding sources, and Hillary, of course, in the station, and answer any questions.